Welcome back to the deep dive. So you, the learner, shared some really fundamental source material with us today. We're looking at key excerpts from Jahangir Moini's Anatomy and Physiology for Health Professionals.
Yeah, this is great stuff. It's really the cornerstone of health science.
It really is. So our mission today is to do a foundational deep dive into, well, the absolute essentials for life.
Exactly. We're going to take these core academic concepts, the structure and function of the human body, and, synthesize them into a clear, high-impact understanding. Something for anyone who really needs to master the basics of A&P.
Absolutely, getting right to the core of how the machine works. So to start, let's just be clear on the language. Anatomy and physiology. What are we studying? And why are they so inseparable?
Well, anatomy is the study of form, the physical structures, the shapes, where things are. Physiology, on the other hand, is the study of function, how those parts actually work.
And they're linked because one dictates the other.
Fundamentally, structure dictates function. The specific shape of a protein determines its job as an enzyme, or the incredibly thin walls of the alveoli in your lungs are perfectly built for gas exchange. You can't have one without the other.
Okay, so if the body is this complex system of structures and functions, it has to have a core operating system. And that brings us to what might be the king of all A&P concepts, homeostasis.
Homeostasis is the single most critical concept.
Yeah.
It's the body's ability to maintain a relatively stable internal environment.
Despite everything going on outside.
Right, despite continuous changes. Your survival depends on this constant, delicate balance. We're talking about regulating everything from nutrient concentration, oxygen levels, CO2, pH, temperature, blood pressure, all of it.
It sounds like a constant battle. And this is where it gets really interesting for me. The mechanisms, the feedback loops. Most people have heard of negative and positive feedback, but is it always just about reversing a stimulus?
That's a really sharp question.
Yeah.
Most of the time, yes. The body relies on negative feedback. It reverses a change to get back to a set point. Think of, blood sugar. It gets too high, insulin brings it back down.
The classic example.
But, and this is your point, sometimes the body intentionally changes the set point. A fever is the perfect example. Your body actively shifts its temperature set point higher to fight an infection.
So it's not a failure of the system, it's a deliberate strategy.
Exactly. It then defends that new, higher temperature using the same negative feedback mechanisms until the infection is gone.
That makes so much more sense. Okay, so if negative feedback is all about stability, what about its opposite? Positive feedback loops just sound inherently risky.
And they are. They are. Positive feedback accelerates the result. It pushes the system further away from where it started.
Further away, so it's not trying to fix anything.
Not at first, no. It's usually for processes that have to be completed very quickly, often under a lot of stress, before stability can return. The classic examples are the powerful accelerating contractions during childbirth.
You don't want those reversing halfway through.
You definitely do not. Or the rapid cascading effect of blood clotting after you get a cut.
Right. And connecting this to pathology, most diseases are basically a failure of this system, a homeostatic imbalance.
That's it. And age is a huge factor. As we get older, our negative feedback systems just become less efficient. But what's really dangerous is when destructive positive feedback takes over. For instance, in some forms of heart failure, the heart's inability to pump causes fluid retention, which then strains the heart even more.
A downward spiral.
A classic destructive positive feedback loop.
Wow, that reveals how powerful those simple rules are. So, if that's the blueprint, let's look at the actual building blocks, the chemistry of life. We have to start with atoms and ions.
We do. So atoms are the smallest unit. You have your electrons, which are negative, protons, which are positive, and neutrons with no charge. For A&P, the most important thing is how they form ions.
Which is all about gaining or losing electrons.
Precisely. When an atom loses an electron, it becomes a positively charged incorcation, like sodium. When it gains one, it becomes a negative anion, like chloride.
And that charge is so important because
the movement of these charged ions across cell membranes is the basis of all electrical signaling in the body. Ionic bonds, the attraction between these opposites, form things like sodium chloride, table salt, which is critical for fluid balance and nerve impulses.
And from those simple atoms, we build the essential macromolecules. Let's start with the big energy carriers, lipids, and carbohydrates.
Right, so lipids. They're carbon-based and they don't dissolve in water. Critically, they are the body's most concentrated energy source. We're talking about 9 kilocalories per gram.
9. And what's it for carbs and protein?
Only 4.
Okay, so that energy density is exactly why the body stores fat for the long haul.
That's it. Now, within the lipids, we have to talk about steroids. These are large, flat lipid molecules. Think cholesterol, estrogen, testosterone. They're vital because being fat-soluble means they can just slip right through cell membranes and act as hormones, regulating homeostasis all over the body.
Okay, and then we have the workhorses of the body, proteins.
The workhorse is a good way to put it. They're the most abundant organic components, maybe 10 to 30% of a cell's mass. And unlike the others, proteins always contain nitrogen.
And they do. Everything. Structure, defense.
Everything. They're structural materials like collagen, they're antibodies for defense, and most critically, they're the biological catalysts we call enzymes. They drive almost every chemical reaction in the body.
So now let's shrink down to the cell itself, the basic unit performing all these functions. What are the key internal factories?
Let's focus on the synthesis and packaging system. First, you have the endoplasmic reticulum, or ER. It's a whole network of membranes. The smooth ER makes lipids like phospholipids and cholesterol.
And the rough ER is where the proteins are made.
Exactly. It's rough because it's studded with ribosomes. And that's where proteins are synthesized and then kind of threaded into these internal spaces, which are called cisterns, for processing.
So the rough ER makes the raw material and sends it to the Golgi apparatus. What's the Golgi's specific job?
The Golgi is the cell's post office. That's the best analogy. It takes the new proteins from the ER, it packages them, modifies them, and sorts them for their final destination.
So that could be outside the cell or somewhere inside.
Right. Secretion out of the cell, or maybe sent to a lysosome for recycling, or even embedded into the cell's own plasma membrane.
And none of this works without power. Which brings us to the mitochondria.
Yes. The mitochondria are the sites of the citric acid cycle, or the Krebs cycle. This is where the cell's fuel is oxidized to produce high-energy phosphate bonds in the form of ATP,
The cell's currency.
the universal energy currency. And when this process goes wrong, the clinical impact is huge.
So what happens when the mitochondria just fail to produce ATP efficiently?
You start to see conditions like mitochondrial disease. It's a whole group of neuromuscular disorders. Because the cells that need the most energy, muscle cells, nerve cells suffer first. So symptoms can include things like weak muscle contractions, heart arrhythmias, even dementia and seizures.
That's a direct line from a tiny organelle failing to a system-wide catastrophe. Okay, let's talk about how the cell maintains its borders, moving things across the membrane.
Maintaining those internal concentrations is constant work. Which brings us to active transport. This is moving substances from a lower concentration to a higher one.
It's going against the natural flow.
Exactly, against their concentration gradient. So it's energy intensive. It requires ATP and specific carrier proteins to get the job done.
And the classic example here has to be the sodium-potassium pump. Why is this thing working constantly just to push sodium out?
Because sodium is constantly trying to leak back in. The pump acts as an antiporter. It kicks 3 sodium ions out for every 2 potassium ions it pulls in. This continuous work maintains that huge sodium gradient outside the cell, which is essential for the cell's resting membrane potential.
Without which, nerve and muscle cells can't fire.
They cannot. It's that fundamental.
Okay, beyond cell, what about water itself? Let's talk about tonicity and osmosis.
Tonicity is all about how a solution's solute concentration affects the volume of a cell. This is life or death stuff in clinical settings, like with IV fluids. If you put red blood cells in an isotonic solution, like 0.9% saline, they're fine. They maintain their shape.
But what if you get it wrong?
If you use a hypertonic solution, say 3% saline, water rushes out of the cells and they shrivel up. That's called crenation.
And the opposite.
In a severely hypertonic solution, maybe 0.2% saline, water rushes in. The cells swell up and can actually burst. That's hemolysis.
Understanding that gradient is literally life-saving knowledge. Okay, let's make a practical connection here between metabolism, energy, and disease. Ketosis.
Yes, this links right back to energy sources. When the body is forced to break down lipids really fast, maybe because of diabetes where glucose can't get into cells, or starvation, or a very low-carb diet, ketone bodies build up in the blood.
And the problem is that they're acidic.
They are. Most are organic acids. So this accumulation causes a rapid drop in blood pH. We call it metabolic acidosis.
And the body tries to fight back, which leads to a very specific clinical sign.
It does. Because the blood is dangerously acidic, the respiratory system tries to compensate. You start hyperventilating, breathing faster and deeper to blow off more carbon dioxide,
To reduce the acid level.
to reduce carbonic acid and raise the pH even just a little. And a classic sign of severe ketosis is a fruity smell on the patient's breath. That's literally acetone, one of the ketone bodies, vaporizing from the lungs.
A perfect example of chemistry forcing a systemic response. Okay, let's scale up from the cell to tissues.
So tissues are just groups of specialized cells. We have four main types: epithelial for covering and lining, connective for binding and support, muscle for contraction, and nerve is for communication.
Let's focus on the support structure for a minute. Connective tissue.
Connective tissue is incredibly diverse. It's bone, cartilage, fat, but also fluid tissues like blood and lymph. Its main jobs are binding, protection, and support. But what's really fascinating is how the failure of just one single protein in this tissue can have catastrophic effects.
You're talking about the thing like Marfan syndrome.
Exactly. Marfan syndrome is an inherited disorder where a glycoprotein called fibrillin is defective. The signs you see are often skeletal. People are unusually tall with long limbs. But the most dangerous effect is cardiovascular.
Because fibrillin is needed for artery walls.
It's crucial for the strength of the aorta. And without it, the aorta can weaken, dilate, and even burst. It's a stark reminder of how important that structural integrity is.
It really is. Okay, let's look at two of the big integrating systems, the nervous system and the integumentary system, or skin.
Right. So the autonomic nervous system, the ANS, controls all our involuntary homeostatic functions. And the vascular system, our blood vessels, is almost entirely controlled by sympathetic fibers. They maintain what's called sympathetic tone.
Meaning they're always a little bit constricted.
Always in a state of partial constriction, yes. This is essential for regulating blood pressure on a moment-to-moment basis.
So if chronic stress causes those sympathetic responses to be overactive, what's the result?
Hypertension, chronic high blood pressure. The vessels are just constantly constricted more than they should be, which increases resistance. That link is a huge clinical concept.
Now, what about our outer defense, the integumentary system? How permeable is our skin?
It's a fantastic barrier, but not perfect. Anything lipid-soluble can get through. We're talking oxygen, CO2, fat-soluble vitamins like A, D, E, and K, and steroid hormones like estrogen.
And some things can make it more permeable.
Right. Penetration enhancers. Alcohol is a big one. It can drastically increase skin's permeability for up to 24 hours after you drink it, which is a huge deal for transdermal drug delivery.
And how does this protective barrier hold up over time?
The signs of aging usually start to show in the late 40s. Skin thins, hair thins, and grays. But the biggest homeostatic problem is with thermoregulation. The blood vessels and sweat glands in the skin atrophy, they shrink.
So it's harder to regulate body temperature.
Much harder. It reduces the ability to shunt blood to the surface, to cool down, or to conserve heat. It's a big reason why elderly people are often much less able to tolerate cold temperatures.
Okay, finally, let's look at the mechanical workhorse's muscle and the heart and how activity versus age affects them.
The key concept here is cardiac reserve. It's the difference between your resting cardiac output and your maximal cardiac output.
How much extra capacity you have on tap.
Exactly. In a healthy adult, it's typically 4 to 5 times your resting output. But in a trained endurance athlete, it can be up to 7 times. It's your heart's ability to respond to stress.
What happens to that incredible capacity as the heart gets older?
The aging heart just becomes less efficient. The valves can thicken and stiffen, a condition called stenosis. This often affects the mitral valve and can lead to heart murmurs.
And the heart rate itself changes.
The maximum heart rate decreases, mainly because the sympathetic innervation becomes less efficient. The heart just can't ramp up as quickly, and the muscle itself gets stiffer, so it fills less efficiently. All of this causes that cardiac reserve to decline. The gap between resting and maximal performance just shrinks.
That was a tremendous tour, from the chemistry of a single ion all the way to the dynamics of the aging cardiovascular system.
To help make it all stick, as the textbook often does, here's a short exercise for you to test your understanding. Compare and contrast the functions of the two major lipids we discussed, triglycerides and steroids. Make sure you include their specific roles and energy yields.
That's an excellent challenge, a great way to connect structure and function. We really hope this deep dive gave you the clarity and the insights you were looking for.
And as a final provocative thought to take with you, we talked a lot about how homeostatic failure causes disease. So consider a very specific example. Bone homeostasis. The body is in a constant balance between depositing new bone and resorbing old bone. What specific disorders like rickets in children or osteoporosis in older women happen when that delicate balance fails? And what's the cellular mechanism that actually tips that scale? There's always more to explore.